All right, so I think so. The first speaker, good morning, everyone. So the first speaker actually, Dr. Nikhilesh Dhar has not yet joined. So we are planning to request Dr. Pudake as he has joined to please deliver your lecture. So for this, what I do is I request Professor Rohan Dhiman to be the panel member of this session. So I request Dr. Dhiman to please take care of this session. Dr. Rohan, please.
Thank you, Professor Vinod Bihari Sahu. And on behalf of Department of Life Science, NIT Rourkela, I welcome Chief Speaker for today's session, Dr. Pudake, and also Dr. Dhar, who will be joining with us shortly. And I also like to welcome all the participants who are joining through Zoom session or through YouTube link and participating in this international conference on omics of plant-pathogen interaction and their application. It's my pleasure to introduce our speaker, Dr. Ramesh Pudake, who is currently working in MIT University. He has completed his PhD from China Agriculture University and has been working at MIT University since 2014. Before that, he worked at Iowa State University and G.V. Pant University of Agriculture and Technology, Pantnagar. He has more than 35 international publications, 3 books, and many book chapters. And he is also serving as reviewer for many international or national journals of repute. He is basically engaged in studying the molecular basis of plant microbe interaction, and he has done research in mechanism of sudden death syndrome in soybean. And this sudden death syndrome is an emerging disease caused by the fungal pathogen Fusarium virguliform. And he has contributed to understanding this fungal infection mechanism through gene knockout studies. After returning from USA, he's been working on biotic antibiotic stress tolerance mitigation through help of beneficial rhizospheric microbes. And as we know that rhizosphere microbiota protects against pathogens, improve growth by producing phytohormones, and may help plants withstand environmental perturbations such as abnormal variation in temperature, drought, and salinity related to climate. It will be really very exciting to learn about this multifaceted beneficial effects of rhizosphere microorganisms on plant health from Dr. Pudake's talk entitled Rhizosphere Biology, Microbes in Action. So I once again, on behalf of our department, welcome Dr. Pudake for joining from MIT University. And we'll be really excited to understand about this field more from his talk. So Dr. Pudake, please.
Thank you, sir, for your kind introduction. And also thank our organizers for inviting me for this conference. Am I audible?
Yes, sir. Yes, sir. Yeah.
Okay. So again, thanks our organizers and giving me an opportunity. So my time slot is changing a little bit, but it's okay. It's I can understand the situations in this pandemic, and it's a great chance to interact with the peoples on this platform. Okay. And yesterday, my mentor from postdoc mentor from Iowa State has explained already in the sudden death syndromes. So I will be not touching that aspect. Let me share my screen. So is it visible to you?
Yes.
Okay. So I will be talking about some aspects which I have work on the rhizosphere coming from the USA, as this presenter has already told that the panelists are already told that I have been working on the rhizosphere biology and trying to find out the microbes interaction with the plant and harvest their benefits for the drought tolerance or the disease resistance. So I'll be touching that topic, and as we know that this will be the outline of my talk, where I will be talking a little bit about the rhizosphere and then we will be talking about the two studies which I have conducted during my tenure at Jivipant University, and also during the uh, project which I have concluded in Amit University under funded by the DST. So these two studies will be shortly described, and we'll try to shed more light on the findings from these two studies which are related to the pathogens. So maybe the omics in fully omics maybe not utilized in these studies. Some aspects are conventionally explored, and that will be explained, and the future prospects of the studies which will be explained in the talk during the talk. So as we know that rhizosphere uh, is of interest in the recent days, and it contains various microbes. Maybe all the microbial genomes or species we can found in the rhizosphere soil, rhizospheric soil. Rhizospheric soil generally is defined as the soil which is attached or which is close to the roots. And another, the soil, rest of the soil is called as bulk soil, and the rhizosphere microbes is the microbes which are present in this attached soil, which may be around a few centimeters away from the root cells, and that contains various microbes and the plant genotypes and the plant root exudates, uh, such as organic acid and small molecules play a very important role in deciding the this microbiome, rhizobium microbiome, and that has an whole impact on the growth and the disease pressures and also other parameters of plant growth. And that is very interesting microbiome which is present in this rhizospheric soil. And most of the studies are being done to isolate microbes which are beneficial, try to understand microbes plant interaction. Many studies have been done on that. Many studies have been done to isolate the biochemicals from this rhizospheric soil, and try to utilize them so that the sustainability can be achieved in the agriculture. As we all know that the problem associated with the plant health can be controlled by the chemical applications in the agriculture, but due to their environmental effect, the day by day the studies are going in the finding out the ways to nullify these effects which are due to the diseases and maybe the abiotic stresses um, through the sustainable approach or the biology dependent approach, and isolate. Many studies has been isolating these bacteria or the fungi which are considered as the bad or the good, and study their mechanism. Generally, this rhizobiome can be classified into three categories of the microbes which are present in this soil. There may be the bad one, which is causing the plant diseases and reducing the crop yield, and causing severe damage to the crop health. So the good one, which will be taking a clue from the root exudates and providing the nutrient supply, providing the plant growth promotions or doing the plant growth promotions by solubilizing the nutrients from the soil. Also, they can be useful for inducing the disease resistance or systemic resistance in the crop plants by providing some kind of hormones or some kind of substances which will be affecting the disease resistance or increasing the disease resistance in the crop plants. But majority of, and the third category I would like to mention is the ugly one, which is also the microbes which are present in the soil, which enter into the human food system and causes some kind of diseases or some allergic effect on the human health. And they will be more dangerous as compared to the bad one, which is causing the plant disease, which can be controlled by various applications. But these ugly pathogens or soil pathogens are maybe causing health hazards in the human. But I'm not talking about the ugly ones, but I will be talking about the good pathogens, which will be helping us to protect our crop plants from bad bacteria or fungi which are present. So many studies in the last decades, or there are many studies for the isolation of the good bacteria or good fungus or endophytes from these rhizophilic soil and utilize them for the plant growth promotions or disease resistance. So many studies focus is on the isolation of the single maybe strain or single species and then using it. But the problem with this single strip isolation, single species isolation is that they may be not effective in the field conditions when they are applied in the diverse diverse soil conditions or the population, the soil containing the diverse population of the microbes, which are native populations. So, which are the laboratory studies which are finding that they are effective for the plant promotions or the say disease suppressions. So they may be not viable or maybe not standing in the field conditions. So the concept of synthetic microbiome was originated, where the combinations of few species or microbial species are inoculated in the soil along with the seed, or maybe added or sprayed on the soil as a synthetic microbiome. So where few species togetherly added, maybe the bacterial species or the fungal species are added in the soil, so that the desirable effect can be achieved. But that also has an competition from the native microbiome which is present in the soil in the field conditions. So we thought of giving some kind of alternative which is available in the nature, and we thought of choosing the compost, which is generally added by the farmers. But we wanted to have a compost which is already selected for the particular characteristics. So for that, we choose characteristics for disease resistance or disease suppression in tomato in vegetable crops. So for this study, we selected various compost. As we know that the compost and disease suppression is a closely associated phenomenon where the microbes and microbial mediated bioactive compounds secreted by this compost microbiome, which is degrading the organic matter into the stable compounds, biochemical compounds, which will be useful, which are useful for maintaining the plant health, also the soil health. So we all know the benefits of the compost, but we wanted to have some kind of selective markers or the selection criteria for the compost, so that the compost which we can amendment, we can add in the soil, should particularly possess some properties, and we should know in beforehand, so that we can add the particular compost, so that the microbiome which is present in the compost will be increasing the disease resistance or disease suppression in the vegetable crops. Also will be increasing the native populations of the microbes present in the soil. So for that we choose various compost. In our study, we chose 32 different composts collected from various localities in Uttarakhand. At that time, I was working in the GB University. So we selected 32 different composts, and we do the bioassay with the pot experiment, where the tomato plants was grown in the compost mix, and compare with the challenge where the vegetables, the tomato plants or the tomato seedlings were challenged with the inoculum of the Fusarium oxidiosporum, which was causing the wilt of the tomato. So we found that various compost has disease suppression capability, and based on the bioassay, we selected or shortlisted 7 composts, which was showing up to 100% of the disease suppression in the seedling bioassay. So we wanted to see that why this compost was made up of different organic waste, and the different composting strategy was also used, because the different composting strategy also decide the microbial population, because at the time of the starting of the decomposition, the microbial population may be different as compared to the final one, because of the different temperatures and the pH, changing pH at different stages of the composting. So the microbial population depends on the composting process, also the ingredients which are used for the compost, making the compost. So we thought of using different composting methods which was used for that one, and also for the different organic matter which was used for that. So mainly the organic waste was from the field, also the Jatropha waste in that Uttarakhand region was very popular at that time, and that was used for the composting. And we thought maybe that Jatropha biomolecules which are present in the plant maybe also maybe maybe helpful for the disease suppression. So we chose variety of the compost, and found that there are different ranges of disease suppression in these samples, and we selected 7 composts based on the high disease suppression ratio, and from the 80% to 100% in some compost, and from 50 to 40 to 60% in some of the other compost, and one is chosen as a control where there was no disease suppression. So we do uh, amplification of 18S ITS region and 16S RNA region for bacterial and fungal pathogens, fungal microbes, and exposed to the DGG PCR, did the DGG PCR, and then separated the bands on the DGG. DGG stands for the degenerating gradient, denaturing gradient gel electrophoresis, where the degenerating pattern of the bands was used for this separation, and the size was the same, but the degenerating pattern based on the sequence was responsible for the separation. So based on this DGG pattern, we try to isolate the bands which are prominently present in the different compost, and then sequence. This is a picture of fungal PCR DGG analysis of disease suppressive compost, and we isolated these bands, and then sequence it and try to find out which bacteria or which fungi is present. So this is also a bacterial DGG PCR DGG analysis for disease suppressive compost. And this when we compare with the QRT-PCR, we found that bacterial population density was much higher than the fungal population density in the compost, which is also an you can say obvious results were there, but the bacterial population was more 25% more, 25 times more than the fungal population. But sorry, and from the DGG and PCR analysis, we found that there is no consistent difference between the disease suppressive compost and control compost in the fungal and bacterial DNA concentration. So we thought that going again for sequence analysis and the phylogenetic analysis. So based on the sequence of information from the distinct bands which we isolated from the fungal population or single ITF gene sequences, we found that most of them belong to the Ascomycota. And on the tree of the fungal sequences, on this phylogenetic tree, the sequences, fungal sequences detected from the disease suppressive compost were grouped separately. So it was like the fungal population which is present in the disease suppressive compost maybe are maybe common, and they are different from the population which is present in the non-suppressive compost. So that one was the one finding. Then based on the phylogenetic analysis of the bacterial population, which was constructed which was done by the 16 distinct bands containing single 16S rRNA gene sequence. Most of the sequence belong to the proteobacteria, and the subphylum of the proteobacteria like gamma and beta proteobacteria were dominantly present in the disease suppressive compost. And this phenomena is observed in the other studies also, which was done maybe in the later years there with the compost or with the soil. There is also another concept is called as disease suppressive soil, and the researchers are trying to find out the disease suppressive soil where the fungal population or the microbial population or the rhizosporic population is helping in the disease suppression of particular diseases in the specific crops, and mixing that soil in the native soil in field conditions, and trying to increase the population of the beneficial microbes, because we know that this many bacteria or fungi from the rhizosphere are unculturable. And when the cultural microbes, which are found in the studies, in the laboratory studies, which are helpful for this disease suppression or maybe the abiotic stress tolerance, maybe get competition from the native populations. So that kind of studies are there being reported recently, that disease suppressing soil being added in the native soil. So this analysis help us to identify some of the genus or species of the bacteria and fungi which are present in this compost, which is showing disease suppression up to 100%. There was no disease symptoms development in the tomato seedlings during the bioassay of our study. So we also did some chemical analysis, because it is we think that these physiochemical properties, like physical biochemical properties, are very useful for the disease suppression, like biological components along with the chemical components by these bacteria or fungi are also helpful. So this we did the compost analysis with the GCMS and we found that benzoic acids, some compounds like benzoic acid, mystric acids, and cholesterol was positively associated with the disease suppressiveness. And the presence of some compounds like monoic acid and glucinol and cytosterol correlated negatively with the disease suppression. So this was according with the earlier studies or some reported studies before. But our main interest was to find out the markers, or to decide the markers which can be helpful us to identify the disease suppressive compost or disease suppressive compost before adding into the soil. We can add, if you can have a marker that will be beneficial, because we don't want to go for the bioassay, and with the help of these markers we can just check the presence of particular bacterial population or particular fungal population in that compost, which will be correlated with the disease suppression. So that one was the aim of our study, and we developed a marker, a PCR-based marker based on the a unique band which was present in the bacterial DGG gel, which was present uniquely in the disease suppressive compost. And we decided to take it as a base and then design the marker. So for that, we again chose a few other compost where we can see how correlate the disease suppression with the presence of the this actinomycetes like bacterium, which was present in our earlier compost. So we selected 7 new compost and done again the bioassay of that compost, and find out that they also show the differential disease suppression. And so percent disease and fresh dry biomass of the tomato plants grown in the compost and wet soil infested with the Fusarium oxysporum was done, and that one the marker design on the sequence information of the selective community like actinomycetes like bacteria was used on the new compost and the old compost, and we can correlate the presence of this band or the intensity of the band with the disease suppression percentage, and it was found that in the new compost and also old compost, the disease suppression was correlated with the presence of that particular bacterial community. So we can say, based on these results, we can say that we have identified a marker which can be useful for identifying the compost, which can show the disease resistance. This is the only study. Then I thought of moving off out the place, and then the study was stopped there. But we can, from our study, that the molecular marker can be a valuable tool for preliminary testing of the compost for disease suppressive nature. And as we know that compost microbiology is a still very complex phenomena, and there are possibilities of involvement of individual microorganism or group of microorganism in disease suppression. So this need a holistic approach where all the parameters of the compost can be need to be studied, and then only the fully benefits of this composting or the adding of the compost can be utilized or can be full extent up to full extent they will be utilized. And they need more studies, and I will be talking about that one in the concluding remarks. But when after that, our interest was focused on the mycorrhiza and other endophytes, which was helping us, which we know that they are helping the plants for the abiotic stress tolerance, also the biotic stress tolerance. So in our preliminary studies, we found that in finger millets, the mycorrhiza infection was differentially regulating the plant growth promotion based on the genotypes, and the plant growth promotion was occurring there. So there is a differential expression of the genes in the finger millet with the when we have inoculated the finger millet with the mycorrhiza. So that make me interested in evaluating this mycorrhiza interaction with the finger millets under the abiotic stress. And why we chose the mycorrhiza and why we chose the finger millet, because we all know that the the millets are very important in the recent days, and many people are preferring the diet with the millets as compared to the conventional cereals, mainly rice and wheat, because of the health benefits, also due to the containing of the various salts which are present in the high amount as compared to the rice and wheat. So they are being preferred for the human consumption. So it is also advisable that we frequently modify our diet with this kind of millets, which will be having a nutraceutical role in human health. So we chose the finger millet, and finger millet is the crop, in the third important crop in case of the millets. When you talk about the millets, this is the third important crop in India, and generally grown in the Uttarakhand region and many parts of India where the dry conditions are present. So we thought of evaluating or exploring the possibility of using the endophytes and the mycorrhiza for abiotic stress tolerance, because in many field conditions, the finger millet is exposed to the drought or the water scarcity. So it is severely affected by various stressful environmental conditions, including the drought. So we know that the symbiotic association of several microorganisms with the plant can elevate the drought stress in the plants. So for that, we chosen the Rhizophyllum intraradiaceae, arbuscular forming a mycorrhizal fungus, and the Piriformospora indica, which is also a mycorrhiza-like endophyte, which is culturable, where the rhizophagus is not culturable, but we can produce the inoculum in the trap crop, and then the spores can be useful for doing the assay and establishing the association with the our taste crop, the finger millet. So we grown it and maize as a trap crop, and that inoculum was used for the inoculation of the finger millet. And Piriformospora is a culturable mycorrhizal like endophyte. So that one also used in a comparison with the Rhizophagus. So we chose 3 treatments where the control and the finger millet was inoculated with the R radiasis, and the finger millet seeds was inoculated with the P indica spores. So, and we chose 3 different water levels where the control plants was water up to the field capacity, and the moderate stress or mild stress was given at the 60% field capacity, and 40% field capacity was chosen as severe stress, where the moisture content was measured, and it was maintained for 20 days, and the stress was given to the crop plants, and the plants was harvested for the biochemical analysis. So this is the methodology schematic of the methodology which we use, where this mycorrhiza was grown in or multiplied into the maize as a trap crop, and that roots and the soil or the soil medium was used as a inoculum, and the P indica was cultured on the hill temper media, and the spores count was measured, and it was used for the treatment with to the seed, finger millet seeds. So when we studied the physiological parameters, we found that both fungus are helping the plant to mitigate the drought stress by increasing the chlorophyll content, also the relative water content in the all the stress conditions. In all the stress conditions, they are significantly improving the performance of the plants. Also, the electrolyte leakage was reduced in the severe stress. In the severe stress are also in other normal and also mid stress conditions when they are infected with the intra-radius or the P indica. Also, the oxidative stress marker also has similar effects on the crop growth of the seedling growth of the finger millets, and we found that the MDA and the H2O2 was reduced in the finger millet inoculated with the this endophyte and the arbusculum AMF under this mild stress and also in the severe stress. Also, the proline content was increased. The total soluble sugar also was increased in this infected plants or inoculated. I will say inoculated rather than the infected plants. And we also did some assay with the antioxidant enzymes and try to find out their presence or their quantity in the inoculated plants and under the various stress conditions. So we found that the supports the antioxidant antioxidant enzyme increase in the finger millet seedlings under the stress conditions. So we concluded that it is a kind of overlapping both the fungus, the Piriformospora indica and Rhizophagus, having some kind of overlapping mechanism where they are providing the nutrients, or they are helping the plant growth by providing some nutrients, also the regulating the enzymatic or antioxidant enzymatic activity by and the promoting the drought tolerance in the finger millet crops. So that one was concluded from the biochemical assay. Further, we did the transcriptomics of these infected plants, but the study was not was stopped due to the scarcity of the fund, or the project was stopped there. But the future, maybe we will be exploring that transcriptomics for the various genes which are upregulated or maybe downregulated in the finger millet due to the inoculations of these two beneficial fungus in the finger millet. So we concluded that study, and we found that these two, both the fungus inoculations, can be helpful for us in mitigating the drought tolerance. So that was the second study we concluded regarding the. But we also will know that this is a very complex system, and many microbes are involved in that one, and also the other criteria like the soil pH, soil organic matter, and other parameters are involved there. So there is a new concept is coming in a scenario that we will have to engineer this rhizosphere by using various or the various approaches, not just by adding 1 or 2 PGPRs or the microorganisms which are promoting plant growth in the soil, but a holistic approach where the genotypes of the crop plants will be selected or decided which genotypes can be chosen for the cultivation in that particular soil, and so also the adding the substances like agricultural inputs so that the health of the soil will be maintained. And then managing the soil microbial populations by adding some kind of supplements. Also the organic matters and compost may be added in that soil so that the beneficial population need to be increased. So for this, achieving this rhizosphere engineering, we need a holistic approach, and we need to study many parameters rather than studying only 1 or 2 parameters, which we have also experienced in our study, that this laboratory studies may be not viable in our field conditions. So we think that there is a further need, and the omics or the omic tools like metagenomics, metatranscriptomics will be helpful in our understanding of this rhizobium microbiome assisted disease suppression, or maybe the abiotic stress tolerance. That will need to be maybe for focus. And many studies are being done, and still, but there are no kind of conclusive evidences based on the, or we can say the was foolproof strategy is not there still date. But this I'm fully confident that these metagenomics the omics tools will be helping us to have a rhizosphere which is a healthy, also promoting the health of the plant under the challenging conditions. So with that, I would like to conclude my talk, and thank you. So I'm open for the questions if you have any questions.
Thank you, sir. It's a nice presentation. So, Rohan sir, please.
Yeah, thank you very much, sir, for the nice talk, and I think so the information that is disseminated through your talk, it will be really very helpful. And now I think so I'll take up questions which are there, I think so, in Zoom platform. So there is a question from Dr. Amrita Panda, and the question is, sir, how you screen the disease suppressive compost?
Okay, so for disease suppressive compost, we mix the chosen compost with the soil. For this, we did the pot experiment where we added 20% of the compost in the soil, which was a sterile soil. Sterile soil means we autoclaved the soil so that the natural or the microbiome which is present there will be killed, and the role of the compost microbiome will be playing important role in that one. So we can differentiate between the disease suppressiveness of the different compost. So we added 20% of the compost and did the bioassay. During the inoculations, during the seedling transplanting, we inoculated the tomato seedlings with the Fusarium pathogen, the spores of the Fusarium. So we allowed it to grow, and then try to find out how much symptoms are produced on the tomato seedlings after particular individuals intervals.
Ramesh, please stop the share the screen.
Okay. Is it okay now?
Yeah.
Okay.
Next question, sir.
And I think so in continuation to our first question, the second question is it completely based on morphological symptoms of tomato plants?
Morphological symptoms, yeah, physical observations of the plant. We haven't checked infection level or some kind of cytological studies were not done, but based on the symptoms development.
Okay, okay. Next question is from Dr. Santosha Rathore, and the question is, sir, which experimental design was used to record the phenotypic data?
Simple CRD was used, completely randomized design, because it was a greenhouse study, and the pot was exposed, kept randomly in the greenhouse conditions. So CRD was used.
Okay. Another question is from Ms. Rujita Mishra, and it is like when you are introducing different soil microorganisms, so whether it is harmful for the soil fertility or not. So that is another question. Yeah.
Introducing soil microbes, particular microbes in the native soil, maybe have another impact on the rhizobial populations in the soil conditions, native conditions. So I don't see any harm in that one, but if you are adding that need to be evaluated, and it depends on the microbes which you are adding, and it depends on the soil conditions. So I don't think there may be a soil fertility or reduction in the fertility, but maybe infecting somewhat negative or positive way on the microbial populations because of the competition, inter-competition between the microbes.
So I have one question, like when you are talking about the rhizosphere engineering, so that will not be same for all the different areas or geographical locations, isn't it? So it means first we have to go through a particular community or that particular area to see what type of engineering methods or what type of, the rhizosphere engineering methodologies we have to use, isn't it?
Yes, it will be a specific to a particular region or particular type of soil, because the chemical composition of the soil will also in maybe having a role on that engineering results, whatever we are doing. Maybe not be. I don't know. Maybe the studies are like need to be done. I think that they need to be done by done at the specific location or specific type of the soils, rather than having a generalized thing, because every soil and every crop has a specific requirement, and also their environment is different, like a root exudate or the quantity of the root exudate, also the pH of the soil. So that are very complex systems. So that need to be evaluated case by case.
Right.
So questions are also coming in YouTube channel, so that we are going live. So Amber Gupta, he asked, like, does season affected the growth of bacteria, and are most bacteria mesophiles in the nature?
No, I don't have any idea about that. Only as seasonal effects, I don't have any idea.
Okay. And one more question. Biswajit Sahu is asking, which type of marker and from which you are isolating the DNA for PCR, and also which type of PCR you are doing?
The question is not clear to me. Which type of markers?
Which type of marker to identify the soil type and the type of PCR?
We use RT-PCR, semi-RT-PCR, also the real-time PCR for measuring this population. So we use universal primers for this ITS regions and 16S RNA regions for the bacterial, for the fungal population and for the bacterial population. So they are universal primers which will be studying, which can amplify all.
So ITS, those markers are established ones.
Already established. They are already established.
Okay. So one more question. Madhusmita Pala is asking, rhizospheres, are they associated with angiosperm or lower plants?
They are associated all plants. Rhizosphere is associated with all the plants. It's not like you cannot say that any plant present on the earth is not associated with the rhizosphere. They are associated with all plants, all category of the plants.
So, sir, I think so from Zoom platform, there are no more questions. So, Bino sir, do you have any more
Yeah,
questions?
I think so. From YouTube also, we addressed all the questions asked by the audience.
Okay, okay. So, as we are aware now that Dr. Nikhilesh Dhar is not joining with us in this particular session. So, because of unforeseen circumstances, he'll be joining with us in tomorrow's second session, which will be starting at 3 o'clock. So, for the time being, I think so we are done with the first session, and we will be meeting again at 3 o'clock today for our second session for as far as today's talks are concerned. Isn't it, sir?
Yes. So we would like to thank Dr. Ramesh for his nice presentation and addressing the gathering with all sorts of the questions. And thank you all for attending the seminar, this first session. So as Rohan sir told, so the speaker, Dr. Nikhilesh Dhar, could not make it due to some technical glitch or something. So he will be attending, so delivering his lecture. So tomorrow, in the second session, 3 p.m. So followed by Rajesh, Dr. Rajesh and Dr. Prashant Singh. But today again will be so joining at 3 p.m. with the scheduled program, and see you in the second session. Thank you all.
Okay. Thank you, sir. Thank you very much, sir. Thank you.
Thank you.